Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you very much for the honor and for the invitation. And thanks a lot to Dr. Muhammad Nashi for explaining a lot of stuff that I should have invested time on. That means that I can literally fly by with all the concepts, and I can directly jump into the conclusions or the main studies or results from our study. So first things first, I would like to acknowledge and thank all the funding agencies, all the collaborators from all over the world, as well as a lot of journals who thought our work worthy of presenting on their front or back covers. Now we know, we have it here, we know that the puzzle actually has been that you generally have to spend $2.6 billion for one drug to go from lab to the market. $2.6 billion is equal to 12 astronauts being sent from Earth to space, or if you have Elon Musk, it comes to 20 astronauts to space, and about 10 to 15 years. The reason, intergenome variability. And in some cases, it can even go to 99.6% failure rates. Why? Because these animals who want to study a particular disease, they don't get the disease in the first place. So automatically, their response will not be the response that you see in humans. That's why we are here. The solution is to actually form these microfluidic chips or organ-on-a-chip devices or 3D bioprinting so that you can actually have a one-to-one recapitulation of human disease in the lab, and that will actually give you enough chances for successful drugs into the market. That's why I actually stand. I go from why to how, and I use a lot of other subjects, for example, biology, chemistry, and engineering, to actually go from a disease etiology to finally find something for therapy. I generally work in the fields of biofabrication, biomaterials, and bioimaging, but in the interest of time, I will quickly pass by only with two different subjects of biofabrication and biomaterials. Biofabrication, as Dr. Muhammad Nashi was already talking about, bioprinting, and you have different types of things. You have different bioprinters. You can actually either extrude things through the bioprinter and then crossing them, and voila, you're there. Or else what you can do is that you can actually use light the way that we are actually using, for example, the overhead projectors, right? So they actually shine things on the white background, and then if you have any photocross-linkable element on that white background, you can either go for a positive or a negative mold. That's exactly what we do here. We actually could make, a phone-based bioprinter or a multiple extrusion bioprinter, or even if you are not really technically savvy, you want to have something which can think for yourself. You don't actually care about, learning the whole technique. What you need to do is that you need to actually have this intelligent bioprinter in the lab that will actually do all the things. It will look at the type of injury there is. It will look at the depth of the injury there is. It will automatically decide also what kind of biomaterial can actually go for that particular injury. So this is the phone-based DLP biofabrication. This was the ray optics of this one. You literally had, iPhone 6, and then I bought something from Amazon which cost us $120. And then based on whatever we actually write on the phone, you can actually, reflect it and focus it, and there is a batch system. You can directly start printing your construct. And we could actually do, different types of architectures. We could go for windows, microchannels, tumor vasculature. This tumor vasculature is taken from an NIH STL file from an actual tumor. And then we could also go down to less than 50 microns to be able to come up with structures that can actually help you go down with the with any kind of diffusion limitation that you generally will have if you want to go for higher or thicker constructs. We could make different types of structures, including some organs. We could make nose, we could make an ear, a kidney, a heart, and a brain structure. But what we went on for just the initial testing was a bone construct, when we could actually make a bone scaffold, put it inside a bone, and for at least 14 days, we could see that there was no cell death. It was actually thriving quite well. There was not also not any fibrotic response or immune response from the animal. And green is life, and red is death. So there is no death. And then it takes me to the next one, where you have intravoxel bioprinter. Imagine that there is a big someone from Mars or some other genus comes in through the door, and he or she or it actually starts to cut my arm. And if you now see the arm from this angle, you'll see a lot of heterogeneity. You will see, first of all, You have bone, then you have bone marrow followed by bone. You have muscle, skeletal tissues. You will have vessels. You will have finally skin. And all these tissues are pretty heterogeneous. So what do you want to do actually if you want to have all these tissues printed together or made together in the lab? We could actually do that with this type of bioprinter that we made in the lab. It could actually print 7 different types of bioinks. And based on the mechanical strength of those bioinks, you could actually have check valves in there to be able to see that you can have a streamlined flow of all the inks. That means that all the different tissues of different mechanical strengths can be made in the lab. What you now see here is this was actually at that time one of the fastest bioprinters in the lab. It could actually generate at least 3 or 4 bioinks quickly in the lab, and we could also make small different types of these blood vessels which can be directly used for perfusion of blood. And what you see here on the left side is a mini liver that's getting printed in the lab, and you could actually also see it inside the lab. After 21 days, these hepatocellular carcinoma cells have a property that they actually form spheroids. So you can see that there are different spheroids formed, and that they were also able to release albumin as compared to the monolayer cells, which was an arm of doing cell culture or doing any kind of tissues. And then here we could actually also on the right side you see an actual bioprinting of a muscle tissue. This one is a God-made muscle. This is the architecture. This is Shabir-made muscle, and this is the architecture of that one. In 21 days, you could see that the myosin heavy chain expression was quite good, and that means that it was going in the right direction. But again, we had to actually publish, so we could not go for the animal studies. But as Dr. Noshi was saying that, we have people like Anthony Atala who actually got last year $100 million of grants. They have enough funds to actually work on the animals. But again, the oral job from NIH and FDA is to minimize the use of animals through the models that we generate generally in our lab. That is the 3D culture or 3D biofabrication 3D printed models, so that you can have one-to-one correlation of what you see in the lab, what actually gets transcribed in the field as well. Now, if you don't want to invest any time in printing or making your own bioprinter, what we did is that we actually also made this intelligent bioprinter. It's a 6-axis robotic arm. It's an intelligent in situ printing guided by eye and hand robotic technology. It could actually look at the color of the skin, the depth of the wound, and it could automatically start either spraying or extrusion based on how the actual injury is, whether it's too much, volumetric, or it's just just on the surface or topical. So you could actually also make different types of structures over there based on the material you have. You have fiber-like. You can make a muscle. If you have pores, you can use it for making vessels or vascularized. And if you have elastomeric substances, you can actually make something like a skin. We could, and it was intelligent enough to be able to see whether or not you have the concave or convex. So it will automatically decide on its own. It will map the whole surface. It'll see what type of surface it is. It can go also at an angle, like 25 degrees Celsius, 25 degrees. It'll automatically treat the different types of, you can see here, it's actually coming at an angle. And with a pig, we could actually also we could make 3 different types of layers, where it was just the skin or the muscle as well. So it could actually really print the muscle tissue as well as the skin. And it also had this kind of thing, like, for example, in a battlefield, if there's a soldier who actually loses their nose or ear, this bioprinter was intelligent enough that it could be trained what type of structure it is that's missing. So that's what we did here. We actually showed this pattern to the bioprinter. Unfortunately, it's a little bit glitchy, but it could actually generate the missing pattern that we took it out for. And then based on this stuff, based on the injury type as well, it can actually go for a dot, boop, or a couple of layers, or a lot of stuff based on the severity of the injury. It can actually go and decide on its own. So in summary, we're able to fabricate, highly resolved different structures that could be used for pathological studies and try to understand how things can work. And at the same time, we could also actually make a bioprinter which was intelligent enough that it did not leave the decision to you. It could actually decide on its own what type of injury is there, what type of tissue is involved, how deep is the tissue, at what angle it needs to actually make. Now, if you do not want to be too technical, if you don't want to actually make bioprinters, what can you do? And if you don't have enough money, to be able to get bioprinters from the market, even though they cost $10,000 to $15,000 in the US, but here it is 300,000 dirham. That's almost 10 times. So you can't buy it. What other innovations can you make from biomaterials to be able to make them full clinical size thick implants that can be implanted without bothering about, number one is diffusion limitation, and number two is the survival of the implanted structure afterwards. That's why the magic begins. But before we go, we want to talk about cooking, bakery. Who bakes things? Raise your hands. Lively, yeah, more. Just these people. Baking, who likes it? Good. So when we talk about baking, we talk about flour, eggs, sugar, and oil, right? That gives us a dough. Then whether we add a chocolate, it gives a chocolate cookie. If we add butter, it gives a butter biscuit. If we add ginger, it gives a gingerbread. And I can assure you, biomaterials is not different. You have a biomaterial here, you have cells, you have an extracellular matrix component, and you have any growth factor. You can make hydrogel. Then based on what you add to it, if you add one particular thing, chemokine, TGF-beta, you get cartilage. If you add bone morphogenic protein, you get a bone. If you add vascular growth factor, you get blood vessels, right? That's precisely what I did. We actually made small particles which are as big as an RBC, 5 to 7 micrometers big. And then based on what you do with them, you can make subcutaneous skin model. If you want to use silicate nanoparticles, you can get a bone. Or if you just directly want to use another chemokine stromal cellular factor, you can actually treat an MI, and myocardial infarction in a rat. So this is how we actually looked at increasing the oxygen generation from these scaffolds. So you now have a thicker scaffold that can generate oxygen on its own. Automatically, you can go for thicker constructs and implants, and you could increase the oxygen release from calcium peroxide by adding it to another polymer, for example, polycaprolactone, so that it can provide oxygen in the preclinical phase, which is more important. And we could actually make skin. You can see that it does actually produce really good blood vessels. And then we could actually also use it for MI. You can see that after some days, we could literally look at the function is back, the cardiac function is back. The ejection fraction is really, really good. The overall volumes of the left ventricle are pretty enhanced. That was after 21 days of inducing an MI in rats. The take home is it's an injectable, oxygenating, silk-based biomaterial attracting stem cells to heal MI injury and enhance functional recovery of the myocardium. And we also used it to enhance the bone formation or the decision to make either bone or a cartilage. And we could actually look at these particles which were oxygen generating. They could actually increase the fate. If you look at the osteogenesis or osteoblast differentiation, these genes, this is the transcriptomic data. You can see that when we use the oxygen-releasing particles, it actually does enhance the expression of the genes which are responsible for the osteoblast differentiation. So we could see that we could actually increase the different types of genes which are responsible for the bone formation. But more important than that, we could actually also look at, for example, different types of injectables that can treat diseases like diabetic retinopathy or acute macular degeneration. Here's a gel which can directly be injected in the eye. It automatically cross-links. You don't need a cross-linker, and it can generate or give release the drugs pretty nicely. It had really good transparency. pH was good, and also the refractive index was on spot. Then comes the concept that Dr. Noshi was also talking about, the decellularized extracellular matrix. We could actually use the goat intestines, decellularize them, use them for deep infectious wounds or deep burn wounds. And we also have some stuff going on for the spinal cord injury. In summary, we treat the diseases that can torn skin, fractured bones, or broken hearts. We just heal them. And we can actually heal injuries you can see, hear, or feel. And thank you very much.
Sure.